started. All right, so test 1 should be finished. Lab exam 1 is finished. We are now starting our next set of material, which is going to be Chapter 22 and Chapter 23. I did put some objectives for the hybrid class. I'll try to make those available to the other classes as well. Some objectives to make sure you know what you're trying to accomplish from these 2 chapters. We are going to not go into the pathologies, and we're not really going to do a lot of the T cell and the B cell specifics, which come at the end of Chapter 22. So try to use the slides to kind of guide you on what you need to really learn and read in each of the chapters. All right, so Chapter 22, lymphoid and immunity. Okay, so there's 2 parts to this organ system. One part of this organ system is you have a system of tubes that are about returning fluid that has been lost as blood moved through the capillaries, because in Chapter 21 we learned hydrostatic and oncotic pressure led to a little bit of loss of blood water, plasma and some ions and some of the small proteins to the interstitial or extracellular fluid around the cells and in tissues. So part of what the lymphatic system is collecting that fluid and returning it to venous circulation. The other side of it is we now have a fluid that's flowing through tubes that we can check and see, do we have foreign objects, pathogens, things that want to do us harm that we can remove and fight against and mount a defense of immunity against. So the main objective is, of course, return the fluid. The immunity side is while we're returning the fluid to do a check and to make sure that we haven't been invaded and we haven't been overtaken by foreign objects. A pathogen is going to be what we describe as a general sense, anything that's trying to come in and do us harm. So bacteria are pathogens. Virus are pathogens, fungus, pathogens, parasites, pathogens, amoebas, protozoans, ticks, worms, they can all be considered pathogens. Their goal, in many cases, is to get into our body system, utilize some of our resources to propagate, grow, and take over, and in doing so, that can either harm us, debilitate us, take us, like malaria doesn't necessarily always kill us, but, it does sicken us. But in many cases, some of these pathogens are going to kill us, okay? So our lymphatic system is a means that we try to, again, keep a check of our body water, because think of it as kind of like a circulating body water fountain. And so as we're, capturing that water and getting ready to dump it back into the circulatory system, we do a check to make sure we haven't picked up any foreign objects. Okay? And the cells that are going to play a role in this system are going to be all of our white blood cells. So we are going to review and go into a little bit more detail what these white blood cells do and how they participate in the non-specific parts of immunity, meaning how do macrophages eat anything that they come across? How do neutrophils, eosinophils, and mast cells, which are basophils, spit and then phagocytose the items that they tried to kill by releasing their granules. And then we're going to go and review some of the other systems such as the integumentary, the digestive, any system that has a epithelial barrier trying to prevent a pathogen from getting into our body, whether they're airborne, foodborne, or contact-borne. Okay? Now, the beauty of our immune system is while we have these kind of and general means set up to keep pathogens out, if a pathogen does get past our first and second tiers of defense, we have a system in place that allows us to then identify and mount specific defenses cell-wise and antibodies, so humoral-wise, that'll let us find that pathogen and kill it and then potentially provide a lifetime of protection against that pathogen if it's ever exposed to us again. Right? And so that is our specific defenses. And we can manipulate artificially the specific defenses and enhance them through vaccinations, through IV antibiotics or general antibiotic drugs that allow us then to potentially not die from a pathogen that prior to vaccinations or antibiotics used to kill a large portion of the population. Okay, so we're going to go through a lot of this material, and while we're not in an immunology class, and remember immunology classes usually span 2 semesters, people get PhDs in immunology, and then they specify they're a bacteriologist or a virologist, so they can even get more specific and just study viruses or just study bacteria. So we're going to just do a lot of the tip of the iceberg. Okay. All right, so when we look at the immune function part, the immune system and our specific and non-specific defenses actually encompass more than just this organ system known as the lymphatic system. Right? Any organ system that has epithelial barriers, that is going to be part of the immune system. So the skin from the integumentary is a barrier to keep things out. It tries to keep ticks out. It tries to keep worms out. It tries to keep bacteria, viruses from getting into our system. The cardiovascular system is giving a ride to white blood cells because they originate in bone marrow and eventually need to get to the dermis of your skin, get to the lamina propria behind epithelial linings of the digestive tract or the respiratory tract or your urinary tract, right? And then, again, those respiratory, digestive, we have airborne pathogens, we have foodborne pathogens, and then we have pathogens that try to get in the back doors through our digestive tract in the anus and in the urinary system, so urinary infections. So, again, we're going to have those systems that are part of the immune system. They're all part of the non-specific immune system because in place are epithelial tissues, cells that are touching each other, cells that are potentially making mucus or things that trap particles so they can't get between the cells or through the cells and potentially then into the connective tissue and into the bloodstream and into other host cells or areas of the body. All right. So let's focus first on the return of fluid to the cardiovascular system. All right, so let's focus first on the lymphatic side where we're taking blood water that escapes from the blood, collects in our tissues, collects around cells, and let's return it to the cardiovascular system. And that system is going to be that the fluid we are collecting in this network of vessels and tubes is lymphatic fluid. And that lymphatic fluid is going to be plasma minus albumin and some of our big proteins. So it's going to be water. It's going to be some of the small electrolytes, sodium, potassium, calcium, chloride, and small organic molecules, some glucose, some amino acids, some nucleic acids, and it might be some small proteins, maybe some of our hormones, some of our other proteins that we have coming into and out of the bloodstream and hanging out outside of a cell. So maybe some insulin, some glucagon, some oxytocin, some ADH, different things like that. Okay, the vessels that are going to collect them are going to be little like funnels that are going to be around capillary networks, and they're going to have open little pores, and they're going to have collection sites and collect that fluid in the extracellular interstitial fluid. And then like veins, they are going to have low pressure, they're going to have valves, and they're going to slowly bring that fluid back to the right and left subclavian veins. And in doing so, they have to overcome gravity. They have to basically try to bring fluid against gravity's forces, that it's under low pressure and they don't have a pump. So like veins, these vessels are going to be very, very kind of low pressure, very compliant, have high capacitance, and they're going to have valves and they're not going to have a lot of muscle. And they're going to use pressure gradients. Okay? Now, in the lymphatic system, there's going to be some special organs that fall into the system because they either play a role in filtering the lymphatic fluid as it's making its way back to the cardiovascular system, or they play a role in where white blood cells grow up, become mature, or they are big kind of areas where white blood cells congregate. Okay? So, the first one we're going to talk about is going to be the thymus gland. Oh, where's my pen? Okay. the thymus gland. The thymus gland actually doesn't do anything related to fluid, bringing back fluid to the cardiovascular system, and it doesn't really have anything to do with your immune system, but it's where T cells, so this is where T cell lymphocytes go to mature. So T cells leave bone marrow and head to the thymus gland. So its big component and piece of the whole lymphatic system is it's where T cells go to become immunocompetent. Because you have immunocompetent T cells now can differentiate between antigens that are self and antigens that are not self. So it is how the body and the specific defenses of our body know that a cell or a protein or something is either self or not self. So the problem with the thymus gland is if T cells are not immunocompetent, we can't then potentially find pathogens and identify them as not self. Or the T cells can't tell that a cell is a self cell, let's say like the Schwann cells wrapped around your peripheral nerves. And then they start killing cells that should be identified as self and left alone, and in doing so cause an autoimmune disease and problem. Okay? All right. The next big organs that we're going to talk about is the spleen. The spleen is not really involved with return of fluid to the cardiovascular system because the spleen actually gets blood supply. There's a splenic artery, and the splenic artery brings blood to the spleen. So the spleen is going to be one of our organs where we have a high amount of lymphocytes hanging out and taking up residence in here, so B cells, T cells. We're also going to have some of our macrophages, neutrophils, and other phagocytic cells taking up residence in the spleen. And these cells are going to do like the cells in lymph nodes. They are going to filter and check for non-self-expressing cells, non-self-expressed proteins, any antigens that are not self. But they're going to check them in the blood supply directly. So any blood-borne pathogen, your best bet to capture it is in the spleen. And then the spleen is also going to be where the macrophages remove old, worn out, and decrepit blood cells, erythrocytes. Okay? All right, you're going to have these enlarged areas of lymphocytes in your abdomen, and they are known as MALTs because they are called mucus-associated lymphoidal tissue. So remember, your I'm blanking. The peritoneum, the cavity that's the serous membrane, the visceral peritoneum and the parietal peritoneum, there's connective tissue behind the epithelial cells forming the serous, making fluid. Behind those, in that connective tissue, we can have lymphocytes, we can have macrophages, and they can kind of collect into this large lymphoid type tissue. And the goal is that this can happen in the serous membranes, this can happen in the connective tissue behind the epithelium of the small intestines or the large intestines, and we just have a large collection of lymphocytes and macrophages that are behind our barriers that then, should a pathogen try to get through because it's coming up from the large intestines or coming past the stomach and into the small intestines, these cells are there to try to capture and destroy. Okay? And with people with Hodgkin's and non-Hodgkin's lymphoma, many cases they have these enlargements of this lymphoid tissue. So they have these big lymphoid masses. That's because these cells in this, these lymphocytes and these macrophages are inappropriately dividing and growing, and they shouldn't. And so it ends up being that they have a big tumor in this lymphoid tissue. Okay? There are going to be smaller little lymphoidal areas right behind the epithelial known as Peyer's patches. And so similar to the MALTs, they are going to be aggregated lymphoid tissues that are going to be collections of cells behind the epithelial lining in the small intestines. And they're called Peyer's patches. Okay? The appendix can be part of this because the appendix is where in the large intestines we have a large collection of lymphoid cells to help, again, overcome bacteria that live in the gut from ever getting into the, past the gut lining and into our bodies and then causing, sepsis and problems. Okay? All right, so basically when we look at lymphoid tissue, we're looking for a large population of lymphocytes. And the lymphocytes are the T cells and the B cells. Normally you're also going to see a large population of macrophages and other phagocytic cells. So some neutrophils, some eosinophils can also be present there. Okay? And the goal is that as we collect fluid, we have stop points in these nodules in these lymphatic organs that then check the blood or check the lymph tissue to make sure pathogens, anything that has an antigen that's not self, from getting and propagating and taking up residence in our body. All right, there's one more organ that we have to talk about that is considered lymph-rich, so lymphocell-rich tissue. That is going to be your tonsils, and you have 3 sets of paired tonsils. They are going to reside in the mouth and the pharynx, and they are going to be right behind the epithelial tissue of the mouth and pharynx, and their purpose is, like these in the digestive tract, if something gets past the mouth, gets past the lining of the airway and the mouth itself, it could then run into phagocytic cells and lymphocytes, be identified as not self, and then lead to an immune response quickly, and specific immune responses quickly. Okay, so those are our organs, and those are our cells and our vessels. Right? So the main functions of this system: produce, maintain, and distribute lymphocytes. So red bone marrow can kind of be brought into this system because that's where some of our lymphocytes are made. Maintenance, so we have the distributing and the maintenance of maintaining our lymphocyte populations. So anything that leads to overproduction of lymphocytes is going to be a problem of the lymphatic system, a type of lymphoid pathogen. So non-Hodgkin's lymphoma and Hodgkin's lymphoma fall into that. Right? Most lymphocytes are produced or mature in lymphatic tissue and or lymphatic organs. That's where the thymus gland comes into play. And then our lymphatic vessels carry lymph from the capillary network of peripheral tissues back to subclavian veins and return it and help return the viscosity of the blood to maybe be a more watery fluid. Okay. All right, so let's look a little bit deeper at the capillaries. Capillaries are going to be located, lymphatic capillaries, almost everywhere we have systemic capillaries. If we have systemic capillaries, remember the arterial has pressure, the artery side of the capillary is under pressure. By the time we get to the venule, there is a net loss of some of the plasma fluid. Because that fluid would collect and cause problems, these lymphatic capillaries can start to collect that excess fluid and return it to the venous system. So they originate as kind of these little pocket areas with open pores that allow fluid to seep in and then collect and be returned to the vasculature in the veins. Right? They have large diameters, like veins, low pressure. They can be irregularly shaped, and they get a special name when we refer to the lymphatic capillaries in our small intestines area. So if this is a picture of the small intestines, then lymphatic capillaries could be called lacteals. And they're going to play a bigger role in the small intestines with absorption of fat-based molecules. And the reason why, blood is moving through capillaries pretty quickly. Blood is water. Water and fat don't necessarily like to mix. So when fat is absorbed from the epithelium of the digestive tract, it doesn't necessarily want to jump into a water-rich fluid. And so it's a little bit easier then to make these kind of large lipid droplets called chylomicrons and then bring them into the body, into the liver, and into circulatory system through the lymphatic vessels. And so because they do that little special bit, they're known as lacteals. Okay, so taking a bigger look at the micro anatomy. So the epithelial cells might cross over a little bit, but they're going to have more pores, more kind of holes that allow fluid to seep in. Right? The basal lamina, so the basement membrane here, is going to kind of be incomplete and somewhat fuse into the extracellular matrix of that tissue. And you don't have lymphatics where you don't have blood vessels. So where you don't have capillaries, you're not going to see lymphatic vessels as well. Okay? Lymphocytes will take up residence in these capillaries, but we're going to mostly see lymphocytes collect in the lymph nodules, these little areas where the capillaries and the larger lymphatic vessels kind of congregate together and allow the fluid to mix with large numbers of lymphocytes, macrophages, and other white blood cells to make sure that the fluid in here is not carrying non-self proteins, non-self pieces and parts of pathogens. Okay? So, looking at how this works, blood capillaries, which are at the highest pressure, flow into venules, and as they do so, because of capillary hydrostatic pressure and capillary oncotic pressure, fluid exits into the interstitial space and becomes part of the interstitial fluid. As that fluid would collect, we don't want it to get too large because that would cause swelling, bloating, and problems. We collect that fluid through lymphatic capillaries, which then become lymphatic veins, flow through lymphatic ducts, meaning little openings that allow lymphatic fluid to be returned to the right and left right and left subclavian veins. All right, now we have two ducts. We have one that's going to dump the fluid into the right brachiocephalic vein, or the right subclavian vein, and we're going to have one that dumps into the left subclavian vein. But they're going to have different names. Okay? Now, the valves are going to be present just like in veins because we don't want to let fluid backflow and remain in the interstitial fluid. Pressure